right now, without your permission and without your attention, a few 100 trillion tiny reactions are happening inside your chest, threading oxygen molecules through your blood, the way a needle threads through cloth, over and over, so quietly that you have never once had to think about it in order for it to continue. Your body is warm chemistry, humming along in the dark behind your closed eyes, and it has been humming since long before you had a name for any of it. Somewhere in a cell in your fingertip right now, a molecule is folding itself into a shape it was always going to fold into. The way a wet towel dropped on a chair will always fall into roughly the same kind of wrinkles, because the physics underneath it leaves it no other real choice. You don't have to do anything. You just have to keep breathing slowly and let the chemistry do what chemistry does. It's worth sitting with that word for a moment, chemistry, because we tend to think of it as something that happens in labs, in beakers, behind safety goggles, under fluorescent light. But chemistry is really just what matter does when it's given the chance. It's water molecules bumping into each other in a puddle. It's the slow rusting of an old nail left out in the rain, iron atoms quietly trading electrons with oxygen atoms, no one watching, no one needing to watch. It's the way salt disappears into a pot of water on the stove, not vanishing but rearranging, its sodium and chlorine ions drifting apart and mingling with the water until the two are one gently swirling thing. None of that requires life. None of that requires intention. It is simply what happens when certain kinds of matter are placed near certain other kinds of matter, and enough time is allowed to pass. And if you go looking for this kind of quiet chemistry, you find it almost everywhere you look, in places that have nothing to do with anything alive. You find it in the air, above you, right now, where nitrogen and oxygen molecules are colliding by the billions every second, mostly bouncing off each other harmlessly, occasionally sparking into something new when lightning tears through a humid summer sky, splitting stable molecules apart and letting their pieces recombine into new arrangements, some of which rain back down to the ground as compounds that didn't exist in the air a moment before. You find it in soil, where minerals slowly dissolve at the edges, releasing atoms that used to be locked inside rock, atoms that drift into groundwater and begin new chemical lives entirely. You find it in the ocean, in hot vents deep on the seafloor, where water heated by the planet's own inner furnace comes churning up through cracks in the crust, carrying dissolved minerals into cold seawater, the two mixing in plumes that look, if you could shine a light down there, something like ink dropped into a glass of water, swirling and billowing and never quite settling. None of this needs a listener. None of this needs an audience. It was happening on this planet for hundreds of millions of years before there was anyone or anything around to notice it. The early Earth, if you could have stood on it, which you couldn't, because there was no breathable air and the ground itself was still cooling and cracking and reforming, would have looked less like a planet and more like a chemistry set the size of a world, left running on its own, with no one there to record the results. Rain fell into shallow pools and evaporated, leaving behind whatever had dissolved in it, a little more concentrated each time, the way a puddle left in the sun leaves a pale ring of minerals behind at its edges after the water is gone. Lightning struck the same patches of ground again and again, because certain landscapes attract it, the way certain trees seem to get struck more than others. Comets and asteroids, which were far more numerous back then, fell out of the sky more often than we can easily imagine now, some of them carrying with them simple carbon-based molecules that had formed out in the cold quiet of space, ferrying those molecules down into Earth's oceans like packages delivered to a door nobody had built yet. So the ingredients were never really the mystery. Carbon is an extremely common atom in the universe, and it happens to be almost absurdly good at forming long chains and rings with itself and with other atoms, the way a particular kind of building brick just happens to click together more ways than any other kind. Hydrogen is the most abundant element there is. Nitrogen, oxygen, phosphorus, sulfur, these are not rare or exotic substances. They are ordinary stuff, scattered through rock and water and air and the space between stars, and under the right conditions, they combine into modestly more complicated things almost on their own, the way flour and water, left alone long enough in a warm kitchen, will start to bubble and change on their own, because wild yeast is drifting through the air and landing where it lands. Scientists have actually tried to recreate small pieces of this ancient, unwitnessed chemistry in glass flasks, sealing off little private versions of the early world, sending sparks through mixtures of simple gases meant to resemble what the young Earth's atmosphere might have contained, and watching, sometimes for days, sometimes for weeks, as a clear liquid slowly turned faintly pink, then reddish-brown, as something new began to accumulate at the bottom of the flask. When they went looking at what had actually formed in that murky liquid, they found some of the same small molecules that, today, our own cells string together to build proteins, the workhorse molecules that do nearly everything inside a living body, from moving a muscle, to digesting a meal, to letting you blink without thinking about it. It was 1953, in a laboratory in Chicago, when this first happened, though of course the Earth itself had already run this experiment on a scale of oceans, not flasks, for a length of time longer than we can meaningfully picture. A human lifetime is nothing against it. All of recorded history is nothing against it. It happened slowly enough that patience itself, in the ordinary sense we use that word, doesn't even really apply. There was simply a very long time, and matter doing what matter does, undisturbed. But here is the part that stays interesting no matter how many times you turn it over in your mind, especially at night, when your thoughts have room to wander without needing to arrive anywhere quickly. Having the right ingredients, and even having them assemble themselves into modestly complex little molecules, is still an enormous distance away from anything we would recognize as being alive. A pool full of amino acids sitting quietly in shallow water is still, chemically speaking, just a pool. It doesn't grow. It doesn't respond if you touch it. It doesn't make more of itself. It certainly doesn't dream, the way you might be about to. Somewhere between simple chemistry happening in water and something that could be called truly alive, something changed. Some threshold got crossed, though crossed is almost too sudden a word for something that likely unfolded gradually, in fits and starts, probably more than once, in more than one place, most of those early attempts leading nowhere at all, fizzling out the way a spark can land on damp wood and simply fail to catch. That gap between ordinary chemistry and living chemistry, between a puddle and a cell, is one of the oldest and most patient puzzles that people have ever tried to think their way into. It's a puzzle made of very small things and very long stretches of time, which is part of why it resists easy answers, and part of why it rewards slow, careful attention rather than quick conclusions. And it's where we're going to spend our time together tonight, drifting gently backward, past your own breathing, past this room, past everything you know, into water that existed before there were eyes to see it, following the quiet, patient trail that chemistry seems to have taken, one small step at a time, toward becoming something that could, eventually, hold a memory, or close its eyes, or listen to a voice in the dark, the way you're listening now. So let's go there properly, not in a rush, the way you'd walk into a dark house you already know well, trusting your feet more than your eyes. To understand where life might have begun, we first have to understand what kind of world it would have been beginning in, and that world, the early Earth, is strange enough that it barely deserves the name Earth at all. It would be more honest to think of it as an ember, a large, slow-cooling ember, drifting around a young sun that was itself still settling into its own long adulthood. Picture the planet just after it finished forming, roughly 4.5 billion years ago, built up gradually from smaller bodies, colliding and sticking and colliding again, the way a snowball grows if you keep rolling it through wet snow, except here the snow was rock and metal, and the rolling was gravity, patient and relentless, pulling debris together over millions of years. All that colliding released heat, an almost unimaginable amount of it, enough that for a long stretch of time the outer layers of the planet weren't solid ground at all. They were closer to something you'd find at the bottom of a furnace, an ocean of molten rock, glowing, restless, covering the whole surface, with nothing recognizable as land or sea or sky in the way we'd know those words now. Geologists have a name for this earliest chapter, the Hadean, borrowed from the old Greek underworld, and it's a fitting name, not because anyone imagines demons down there, but because it captures the mood of a world so hostile, so unfinished, that for a long time scientists simply assumed nothing interesting could have happened there at all, just heat and rock and time passing without anything to notice it passing. That assumption has softened somewhat in recent decades, but the Hadean still deserves its reputation. This was not a gentle beginning. Slowly, though, and slowly is really the operative word for almost everything in this story, the surface began to cool. Not evenly, not gently, but the way a bowl of soup left on a counter eventually stops steaming, first at the edges, then gradually toward the middle, heat escaping into the surrounding cold of space, a little faster than the planet's interior could replace it. As the outer rock cooled below its melting point, it began to solidify, forming a thin, dark crust across parts of the surface, something almost like the skin that forms on hot milk if you leave it standing, thin and fragile at first, easily broken, easily swallowed back down into the liquid beneath. Underneath, the planet stayed molten for a very long time, and in some sense it still is. If you could dig down far enough today, you would still find that ancient furnace running, more subdued than it once was, but never fully extinguished. We know a surprising amount about this period from something almost absurdly small. Tiny crystals of a mineral called zircon, some of them barely wider than a grain of sand, discovered embedded in much younger rock in a remote, sun-baked stretch of Western Australia known as the Jack Hills. These crystals are stubborn things, chemically speaking. They resist weathering, resist heat, resist almost everything the intervening billions of years could throw at them, and so a handful of them have survived, quietly, since very close to the beginning, carrying inside their structure a kind of chemical diary of the conditions under which they formed. When scientists in the early 2000s finally coaxed information out of these crystals, what they found surprised almost everyone. The chemistry suggested that some of these zircons crystallized in the presence of liquid water, and that the temperatures at the time, at least in places, might not have been quite as hellish as the name Hadean implies. Not gentle by any modern standard, but perhaps not universally molten either. A world of pockets and patches, hot in some places, less hot in others, an early crust that may have floated, however precariously, over cooling rock, in something a little closer to a planet than a furnace. This matters for our purposes tonight because it means water, in liquid form, may have existed far earlier than people once assumed, possibly within only a few 100 million years of the Earth's formation, which sounds like an enormous span of time, and yet is astonishingly early in the planet's story. Where did that water come from? This is one of those questions where honest scientists will tell you they're not entirely sure, and where more than one explanation is probably true at once. Some water likely came from inside the planet itself, chemically bound up in the rock and minerals that built the Earth in the first place, released slowly as volcanic activity vented gases from deep underground, the way steam escapes from the crack of a lid on a pot that's been simmering far too long. And some water, many researchers believe, arrived from outside, delivered by comets and by water-rich asteroids that struck the young Earth during a period when the inner solar system was still a chaotic shooting gallery of leftover debris, all of it still finding a stable orbit, or failing to, and crashing instead. Which brings us, gently, to the impacts, because this early Earth was, quite simply, under constant bombardment. Not the occasional dramatic collision you might picture from a film, but something more like standing in a hailstorm that lasts for hundreds of millions of years, punctuated every so often by a hailstone the size of a mountain. Some of these impacts were almost certainly large enough to vaporize any ocean that had managed to form, boiling it back into the atmosphere as steam, only for it to rain back down again once things cooled, over and over, in a cycle that must have repeated more times than we could ever count. It's a strange thing to imagine, an ocean forming and evaporating and reforming, again and again, patient in a way that only geologic time can afford to be. The atmosphere above all this was nothing you or I would recognize as air. There was no oxygen to speak of, not in any meaningful amount, not for a very long time yet. Instead, the earliest atmosphere was likely a mixture drawn from volcanic outgassing and cometary delivery, probably rich in carbon dioxide, nitrogen, water vapor, and smaller amounts of other gases, a heavy, close atmosphere, more like the air inside a greenhouse left sealed too long in summer than anything resembling a walk outdoors on a clear day. Whether it also contained significant amounts of methane, ammonia, and hydrogen, the specific mixture that early chemists once imagined when they pictured the origin of life, remains genuinely debated among scientists today, and will sit with that uncertainty honestly, rather than pretending the recipe has been settled. Above that heavy, churning air, the sun itself was a different sun than the one that will rise again in a few hours over wherever you happen to be lying right now. Astronomers describe the young sun as fainter, dimmer than today's, by perhaps as much as 30%, a curious fact that creates its own small puzzle, because a fainter sun should have meant a colder Earth, likely one frozen solid, and yet the geological evidence suggests liquid water was present anyway. Scientists call this the faint young sun paradox, and one of the leading explanations is that heavier concentrations of greenhouse gases, particularly carbon dioxide, trapped enough heat to keep the planet's surface from freezing over entirely, working like an extra blanket pulled over a bed to compensate for a colder night. And through all of this, storms, actual storms, lightning splitting a thick, humid sky again and again, over churning volcanic plains and shallow, steaming seas, in an atmosphere with no ozone layer yet to filter the sun's harsher ultraviolet light, which reached the surface far more directly than it does now, unfiltered, insistent, day after day. It's into this setting, cooling but still restless, watered but still uncertain, sunlit but by a dimmer sun, struck constantly from above and rumbling constantly from below, that we have to place the very beginning of our story, because whatever happened next happened here, in exactly these conditions, and not in some calmer, tidier world we might have preferred to imagine instead. So how do we actually know any of this? lying here in the dark, with the actual events themselves separated from us by roughly 4 billion years, longer than the distance between now and almost anything else we might try to picture. The truthful answer is that scientists don't have a window into that time. There's no photograph, no eyewitness, no single rock that says, plainly, life began here on this day. What they have instead is something more like a very old, very damaged letter, torn in places, faded in others, rewritten by later hands more than once, and they have learned, patiently, over generations, how to read what's left of it. The letter, in this case, is written in stone. Rocks are not just scenery. To a geologist, they're closer to sediment settling at the bottom of a long, slow argument, layer upon layer, each one a record of the conditions under which it formed, the temperature, the chemistry of the water or air nearby, sometimes even the presence of something alive. Most of Earth's oldest rocks have long since been melted, folded, and recycled back into the planet's interior by the same restless geological churning we talked about a moment ago, so almost nothing survives from the very earliest chapters, but not quite nothing. In southwestern Greenland, in a formation called the Isua Greenstone Belt, and on a small nearby outcrop called Aquileia Island, there are rocks that have been dated to somewhere around 3.7 to 3.8 billion years old, among the oldest that have survived recognizably at all. And within some of these rocks, scientists have found something curious in the ratio of two forms of carbon. Here's the idea, and it's worth sitting with slowly, because it's one of the cleverer tricks in this whole detective story. Carbon comes in slightly different varieties, called isotopes, atoms of the same element that carry a little more or a little less weight than their siblings, because of extra neutrons tucked into the nucleus. Two of these varieties, carbon-12 and carbon-13, both show up in the natural world, but living things, when they build themselves out of carbon, show a mild preference. Given the choice, the chemistry of life tends to favor the lighter isotope, carbon-12, pulling it into biological molecules slightly more often than the heavier carbon-13, the way a person reaching into a bowl of mixed nuts might, without quite noticing, tend to pick out the almonds first. Over millions of years and countless generations, this small preference leaves a signature, a slight enrichment of carbon-12 relative to what you'd expect from purely non-living chemical processes. And in some of those ancient Greenland rocks, faint traces of that signature seem to appear, carbon locked into the stone with a ratio that looks, to many researchers, more like the fingerprint of biology than the fingerprint of ordinary rock-forming chemistry alone. It's important to say, gently, that this evidence is contested, and reasonable scientists disagree about how to interpret it. The rocks in question have been squeezed, heated, and deformed so many times over billions of years that some researchers argue the carbon signature could have been altered or even created by non-biological processes entirely, high heat and pressure doing on their own what living cells usually do. Nobody is lying to you here, or to themselves. This is simply what the edge of scientific knowledge looks like up close, less a solid wall and more a hazy border, where the evidence is real, but the interpretation is still being carefully, patiently argued over, the way scholars might pore over a smudged sentence in an old manuscript, agreeing on most of the letters but disagreeing gently on the final word. Slightly younger rocks give scientists firmer footing. In Western Australia, in a region called the Pilbara Craton, there are rock formations roughly 3.4 to 3.5 billion years old that contain structures called stromatolites, layered, dome-shaped mounds that form, in the present day, when mats of microorganisms trap and cement fine sediment, layer by layer, the way a very slow, very patient bricklayer might build a wall one thin course at a time, over centuries rather than days. We can watch living stromatolites forming today, in a handful of unusually salty, unusually still places in the world, most famously in Shark Bay, also in Western Australia, and by comparing their shape and internal layering to the ancient fossilized versions, scientists have built a reasonably confident case, though again, not a completely unanimous one, that these ancient mounds also record the activity of early microbial life, colonies of tiny organisms quietly, cooperatively. There is also, alongside the rocks, an entirely different kind of evidence, one that doesn't come from the ground at all, but from inside every living cell on Earth today, including the ones quietly keeping you breathing right now as you lie there. Every living thing that has ever been studied, from the bacteria in a drop of pond water, to the whale sliding through a cold ocean, to the person listening to these words, shares an enormous amount of underlying machinery. The genetic code, the particular set of rules that translates the information stored in DNA into the proteins that do the actual work of being alive, is, with only minor variations, the same code used by essentially all known life. The molecular machines that read that code, tiny structures called ribosomes, are built along strikingly similar lines in a bacterium and in a human cell, even though those two kinds of cells look almost nothing alike and haven't shared a common way of life in billions of years. And the basic method most cells use to store and release usable energy, a molecule called ATP, ferried around and generated by remarkably similar enzymes, shows up again and again in organism after organism, kingdom after kingdom, as though every branch of the living world had inherited the same old toolbox from a common workshop, then gone on to use those tools for wildly different projects. This shared inheritance is one of the strongest pieces of evidence that all known life descends from a single starting point, a hypothetical ancestral organism, or more likely, an ancestral population of organisms that scientists call LUCA, the last universal common ancestor. It's worth being careful about what that name does and doesn't mean, especially at this hour, when it would be easy to let the phrase carry more weight than it should. LUCA was not the first living thing. It was not the origin of life itself, and it almost certainly wasn't a single, solitary cell drifting alone in an empty ocean, the way the word ancestor might make you picture, a lone figure at the root of a family tree. LUCA is better understood as a horizon, the farthest point back that comparing life today can actually see, the last common meeting point before the branches split apart into the deep, separate lineages that eventually became bacteria on one side and archaea and, later, more complex cells on the other. Whatever came before LUCA, however many earlier, cruder, less successful forms of proto-life might have existed and quietly failed, leaving no descendants at all, is invisible to this particular method, the same way a family historian, however skilled, eventually runs out of names in the parish records and has to stop, not because the family didn't exist further back, but because the paper trail simply gives out. So there are, in truth, two different kinds of darkness in this story, and it helps to name them clearly rather than let them blur together. There's the darkness of deep time itself, 4 billion years of rock erased, melted, and rewritten by the planet's own restless geology, leaving scientists with only scattered fragments, a torn letter with whole pages missing. And there's a second darkness, further back still, past even the last common ancestor, into the true beginning, where no living descendant survives to carry a memory of it forward at all, biologically or otherwise. It's in that second, deeper darkness, past Luca, past the shared machinery, past even the oldest disputed rocks, that the actual question of how life began has to be approached differently altogether, not by tracing descent backward, but by starting instead with plain chemistry, ordinary atoms, and asking, slowly, patiently, what they might have been capable of on their own. Before going any further into that chemistry, though, it helps to pause on a question that sounds simple and turns out to be surprisingly slippery. What exactly is life? Not life as a feeling, not life as a mood on a quiet night, but life as a scientist might try to pin it down, precisely enough to know it when they see it, and precisely enough to know when they don't. You might expect, given how much time humans have spent thinking about biology, that there would be a tidy definition sitting somewhere in a textbook, a short sentence that separates the living from the non-living as cleanly as a line drawn in sand. There isn't. Ask a dozen biologists to define life, and you will very often get a dozen different answers, each one reasonable, each one incomplete in its own particular way, each one leaking at the edges when you push on it too hard. This isn't a failure of science. It's more like trying to define a family by naming a single trait everyone in it shares. You might reach for eye color, or height, or the shape of a nose, and every time, someone in the family breaks the pattern, and yet the family is still, unmistakably, a family. Life is like that. It doesn't come with one essential ingredient. It comes with a cluster of features that tend to travel together, overlapping and reinforcing one another, none of them sufficient on its own, and no single one of them absolutely required in every case anyone has ever managed to imagine. So instead of a definition, it's more honest, and, frankly, more useful for falling asleep to think of a short list of things life tends to need, almost like a checklist that gets rougher and blurrier the closer you look at it, but that still gives you something solid to hold on to in the dark. The first thing is a boundary, a cell, in the loosest sense, before it's anything as organized as what swims under a microscope today, needs some kind of skin, some membrane, some wall between inside and outside. Think of it the way you might think of a house on a cold night. Without walls, heat just drifts away into the surrounding air until inside and outside are the same temperature, and there's no longer any meaningful difference between the two. A house holds warmth in, holds the useful things in, keeps the wind and the water out, and in doing that simple, almost passive act of separating one space from another, it makes a kind of difference possible that couldn't exist otherwise. Early on this planet, long before anything you'd call a cell existed, certain fatty molecules, quite unremarkable on their own, had a curious tendency, when they were surrounded by water, to arrange themselves into tiny spheres, their water-fearing tails all turning inward away from the water, their water-loving heads all turning outward to meet it. Nobody designed this. It's simply what those molecules do, the way oil beads up on a wet countertop rather than dissolving into it, following nothing more than ordinary physical preference. And once you have a sphere like that, however crude, however leaky and imperfect, you have a boundary, an inside distinct from an outside, and that distinction is the first quiet requirement on the list. The second thing is energy, or more precisely, a flow of energy, because a boundary by itself is just an empty container, and an empty container doesn't do anything at all. Life, whatever else it is not static. It is a process, closer to a flame than to a stone, and a flame needs fuel moving through it, being consumed, being transformed, releasing energy that does work before it dissipates as heat and drifts away. Living systems work in something like that same way, pulling in energy from somewhere, whether that's sunlight or heat rising from a crack in the ocean floor, or chemical differences between one pool of water and another, and using that energy to keep themselves organized, to keep building the very structures that would otherwise fall apart. This matters more than it might first seem, because left alone, everything in the universe drifts naturally toward disorder. The way a warm cup of tea left on a table will always cool down and never spontaneously heat itself back up. The way a sandcastle erodes and never assembles itself from scattered grains. Staying organized against that constant pull toward disorder costs something. It always costs something, and that cost has to be paid continuously, in a steady trickle of energy flowing through the system, in and out, the way a river keeps its shape not because water sits still inside its banks, but precisely because water is always moving through it. The third thing, and this is where it starts to get more interesting, is information. Not information in the sense of a book on a shelf, but information in the sense of instructions, a kind of memory that can be read and acted upon. In every living thing known today, this information is carried by long molecules, DNA in most cases, whose sequence of building blocks spells out, in a chemical alphabet, how to build and run the rest of the organism. But information doesn't have to look exactly like DNA to count as information in this broader sense. What matters is that there's some pattern, some sequence, some arrangement that can be copied, that can be passed from one generation of structures to the next, carrying forward not just matter, but a set of instructions for what to do with that matter. Which leads naturally to the fourth thing, replication, the ability to make more of yourself, or more precisely, to make more of the pattern that you carry. A fire can spread, in a sense, catching new fuel and growing, but a fire doesn't copy detailed instructions from one flame to the next. It just consumes whatever's near it and doesn't discriminate. Life is different. It copies, imperfectly but persistently, a set of instructions, so that whatever made this particular arrangement of matter successful once has a chance of being tried again, in a new copy, somewhere else, sometime later. And that word imperfectly is doing a great deal of quiet work in that sentence, because it points toward the fifth thing on this loose list, which is heredity paired inseparably with variation. Heredity means that copies resemble their originals closely enough that useful patterns aren't lost the moment they're made. But if copying were absolutely perfect every single time, with no variation at all, nothing could ever change, and nothing could ever improve, or adapt, or explore a new possibility. Every copy would simply be an exact echo of the one before it, forever, unchanging as a repeated recording. It's the small errors, the tiny slips introduced in copying, that create the raw material for something new to be tried out, tested against the world, kept if it helps, and lost if it doesn't. Which brings the list to its sixth, and, in a way, its capstone requirement, evolution itself, not evolution as some far-off, grand unfolding of new species over millions of years, though it is certainly that too eventually, but evolution in its most stripped-down, mechanical sense. Given a population of things that make imperfect copies of themselves, and given that some of those imperfect copies happen, purely by chance, to be slightly better at surviving or replicating than others in whatever environment they find themselves in, those better surviving variants will tend, on average, over time, to leave more copies behind than the others do. That's it. That's the whole engine. No intention required, no foresight required, no plan required. Just boundaries, holding a difference from the world, energy flowing through to sustain that difference, information stored and copied with occasional small errors, and a patient, indifferent sorting process that lets some variations persist a little more often than others. Put all of that together, a boundary, an energy flow, stored information, replication, heredity, and variation feeding into evolution, and you have something that starts to look, unmistakably, like life, even before it looks like anything you'd recognize as a cell, let alone as a living creature swimming through ancient water. And what makes the next part of this story so strange, so quietly astonishing when you actually sit with it, is that every single one of those requirements, every item on that loose and imperfect list, can in principle be satisfied by nothing more than plain chemistry, ordinary atoms bound by ordinary rules, with no spark of anything mysterious required at all, which is exactly the ground this story needs to stand on before it can take its next slow step backward, into the water itself. So let's take that next slow step backward, all the way to a night in Chicago in the spring of 1952, because it's as good a place as any to start feeling, rather than just being told, what plain chemistry can do when nobody is watching it closely. A young graduate student named Stanley Miller had an idea he couldn't quite let go of, an idea his advisor thought was a bit reckless for a first project, and his advisor's name, in case it means anything to you, was Harold Urey, a man who had already won a Nobel Prize for discovering a heavier form of hydrogen, and who by then had turned his curiosity toward the early solar system and the air that early Earth might have breathed. Urey suggested Miller try something safer, something more likely to produce a tidy result in time for a dissertation. Miller, strong-willed in the quiet way that some young scientists are, asked for a few months to try his own experiment instead, and Urey, to his credit, let him. The idea was almost embarrassingly simple to describe, even if it was fiddly to build. Take a sealed loop of glass tubing and flasks. In one flask, put water and heat it gently, so it simmers and sends up vapor, the way a kettle left low on a stove might fill a kitchen with warmth and moisture. Let that vapor rise into a second, larger flask, and fill that flask with a mixture of gases meant to stand in for the very early atmosphere as scientists in the 1950s pictured it. Mostly methane, ammonia, and hydrogen, with the water vapor mixed through them, an atmosphere with almost no free oxygen at all. Since oxygen as we breathe it today is largely a much later addition, produced by living things themselves, which is itself a strange and lovely fact to sit with for a moment, that the air in your own lungs right now is in large part a kind of biological exhaust, a signature these small ancient organisms left across an entire planet. Into that swirling flask of gases, Miller inserted 2 electrodes, and across those electrodes he sent a continuous spark, again and again and again, hour after hour, meant to mimic lightning flickering through a young, restless sky. Below, a condenser cooled everything back down, turning vapor to liquid, sending it trickling into a small pool of water at the bottom, a miniature ocean waiting quietly to receive whatever the sparks and gases might make. And then he waited. He let the apparatus run, sparking and simmering and condensing, for about a week, and by the end of that week, the water in the little flask, once clear, had turned a pale, cloudy pink, then gradually deepened toward a murky brownish red, almost like weak tea left steeping too long. When Miller analyzed what was floating in that liquid using a method called paper chromatography, essentially letting a sample creep slowly up a strip of absorbent paper so that different molecules separate out by how far and how fast they travel, he found something that made Urey, cautious as he'd been at the outset, deeply excited. Amino acids, several of them, glycine, alanine, and others. the very same category of molecule that living cells today string together, one after another, into long chains to build proteins, the workhorse molecules that do nearly everything a cell needs done, from speeding up chemical reactions to holding a shape steady to ferrying oxygen through blood. Nobody had put life's building blocks into that flask. Nobody had guided the spark, or steered the gases, or coaxed the pink water toward biology. Amino acids had simply appeared, unbidden, out of methane and ammonia and lightning and time, as though the raw materials of life were not some rare, precious secret at all, but something ordinary chemistry falls into fairly readily, given the right stage and enough patience. The result made headlines. It made Miller, still a graduate student, briefly famous in a field not accustomed to fame, and it planted, in the public imagination and in the minds of many scientists too, a comforting picture that the ingredients of life were easy, that a bit of lightning through the right primordial soup was nearly all it took, and that the harder mystery lay somewhere further along the road. But science, done honestly, tends to complicate its own tidy stories, and this one was no exception. Over the following decades, as researchers studied ancient rocks more carefully, as they modeled the chemistry of early volcanic outgassing and the behavior of the young sun, a different picture of the early atmosphere began to take shape, one far less generous to Miller's original recipe. Instead of that rich, reducing mixture heavy with methane and ammonia and hydrogen, many scientists now think the early atmosphere may have been dominated by more sluggish, less reactive gases, largely carbon dioxide and nitrogen, closer in spirit, if not in exact composition, to the atmosphere of Mars today than to the version bubbling inside Miller's flask. Run the same spark experiment through that more reserved mixture, and the yield of amino acids drops sharply, sometimes to almost nothing at all, which is an uncomfortable result to sit with if you'd grown fond of the lightning and soup version of the story. And so scientists, being scientists, didn't discard the puzzle. They went looking for it in other places. Some found reason to think the early atmosphere may not have been uniform, that certain regions, near volcanic vents spewing gases fresh from the planet's interior, or in the churning clouds thrown up by the enormous, relentless bombardment of asteroids and comets that battered the young Earth, could have held brief, localized pockets closer to Miller's reducing recipe, tiny reactive neighborhoods within an otherwise quieter sky. Others turned their attention away from the atmosphere entirely and looked instead toward the rocks falling from space themselves, and this is where the story takes a turn that still, honestly, gives me a small shiver whenever I think about it clearly. In September of 1969, a meteorite fell to Earth near the town of Murchison in Australia, breaking apart as it streaked through the sky, scattering fragments across paddocks and roads, still warm in places when people found them. When scientists later analyzed pieces of that meteorite in the lab, they discovered it was carrying, locked inside its stony matrix, dozens of different amino acids, some identical to the ones found in living cells on Earth, and others, strange and unfamiliar, that no known biology on this planet uses at all. That detail matters enormously, because it strongly suggests these amino acids were not the product of anything alive, not some hitchhiking contamination from a life form we already know, but rather a genuinely independent chemistry, cooked up in the cold dark of space, or inside the churning interior of some long-vanished asteroid, entirely apart from Earth and entirely apart from any living thing. Since Murchison, other meteorites and comet samples brought home by patient robotic missions have told a similar story over and over. Sugars have turned up in some, including simple forms of ribose, the very sugar that forms part of the backbone of RNA. Nucleobases have appeared too, the flat, ring-shaped molecules like adenine and guanine that, joined to sugars and phosphate groups, spell out the genetic alphabet every living thing on Earth still reads today. None of this chemistry required a planet at all. It seems to have been happening quietly across the galaxy, in the cold gas clouds where stars are born, on the icy surfaces of comets tumbling far from any sun's warmth, chemistry proceeding patiently on its own schedule, indifferent to whether any planet was even watching. Which leaves us with a picture considerably richer and a good deal stranger than the single spark inside Miller's flask. Perhaps some of life's earliest building blocks were made right here, brewed up in warm ponds or volcanic pools, or the reactive margins of an early ocean, however exactly that atmosphere was actually composed. And perhaps others rained down from the sky, carried inside rocks and ice that had drifted for millions of years through the frozen space between stars, arriving here already assembled, needing only the right conditions to be gathered up and put to use. Both stories may be true at once, each contributing its own share of the ingredients, and it's that very question of how those ingredients, however they arrived, might have first come together into something like water, into pools and pores, and the close, sheltering chemistry of a very particular kind of place, that the story is about to slow down and settle into next. Let that particular place be the deep sea floor then, some 2.5 miles beneath the sunlit surface, in a darkness so complete and so permanent that no eye, had one existed yet, would ever have found any use there. In 1977, a small submersible called Alvin, barely large enough for 3 people to sit knee to knee inside a titanium sphere, descended into this darkness along a stretch of ocean ridge near the Galapagos Islands. The scientists aboard were geologists, mostly, hoping to study the rocks of the spreading seafloor, the slow rift where 2 plates of the Earth's crust pull apart, and new ocean floor wells up to fill the gap. What they found instead, glimpsed first through a small porthole and then on grainy video, was something almost nobody had expected. Towers of shimmering, mineral-laden water rising from cracks in the seafloor, warm in places and scalding in others, surrounded by a crowd of animals nobody had ever properly catalogued before. Tube worms taller than a person, blind white crabs, clams the size of dinner plates, all of it thriving in a place that, by every assumption of the time, should have been an empty desert, since no sunlight reached down there to power anything through photosynthesis. Something else was feeding this whole community, and it took some patient chemistry to work out what. The answer, it turned out, was the water itself, or rather the enormous difference between 2 kinds of water meeting in the same narrow space. Seawater near the bottom of the ocean is cold, a few degrees above freezing, and it carries dissolved within it a modest amount of oxygen and carbon dioxide, along with sulfate and other ions, the ordinary chemical furniture of the sea. But at these vents, that cold water was being drawn down through cracks in the crust, sometimes for hundreds or even thousands of feet, into rock still warm from the planet's own interior heat. As it traveled downward, it reacted with the minerals around it, stripping away sulfur, dissolving metals like iron and manganese, losing its oxygen entirely, until by the time it re-emerged through the vent, it had become something almost unrecognizable. Hot, sometimes well past the boiling point of water at the surface, though the crushing pressure at that depth kept it liquid. Rich in dissolved metal sulfides, utterly without oxygen, and when this transformed fluid met the cold, oxygen-bearing water of the open ocean, the two didn't simply blend quietly together. They reacted, sometimes violently enough to precipitate clouds of black mineral particles on contact, which is why the hottest of these vents came to be called black smokers, plumes of what looked like industrial smoke billowing upward from chimneys of their own mineral making. some of them taller than a 10-story building, built up flake by flake over centuries as the dissolved metals came out of solution and hardened into rock. It's worth sitting with that image a while, since it turns out to matter a great deal for the question of how life might have started. What you have at a vent like this is not a single environment, but a boundary between two very different ones, pressed together in the same small volume of space. Hot meeting cold, oxygen-free meeting oxygen-rich, acidic meeting more neutral, metal-laden meeting metal-poor. Wherever such a boundary exists, there's a gradient, a difference in chemical conditions across a very short distance, and a gradient like that is, in a real sense, a source of energy, the same way a difference in height between two pools of water can be tapped to turn a mill wheel, even though neither pool by itself is doing anything at all. Living cells today still run on gradients of exactly this kind. Every cell in your body, right now, as you lie there listening, is pumping charged particles across a membrane to build up a difference in concentration on either side, then letting those particles flow back through a tiny molecular turbine to make the energy currency the cell needs to do anything at all, to move, to build proteins, to think the thoughts that led you to put this recording on in the first place. It's a strange thing to consider, that the electrical hum keeping you alive at this very moment traces back, if certain scientists are right, to a chemical trick invented at the bottom of an ancient ocean, long before there was any brain around to have a thought at all. The chemistry of the vent water itself offered something else too, something more like raw material than energy. Iron and sulfur, dissolved out of the rock and carried up in that hot fluid, have a habit of combining readily with one another, forming small clusters, sometimes just 4 iron atoms and 4 sulfur atoms arranged in a tight little cube. These clusters aren't alive in any sense. They're simple minerals, closely related to the mineral called pyrite, which people have sometimes called fool's gold for its metallic shine. But they turn out to be remarkably good at one particular job, which is shuttling electrons from one molecule to another, nudging along chemical reactions that would otherwise happen far too slowly to matter. And here's the striking part, the reason so many scientists have found this whole picture so compelling. Almost every living cell on Earth today still uses iron-sulfur clusters, nearly identical in structure to the ones that form spontaneously in vent water, buried deep inside some of its oldest and most essential proteins, the ones involved in extracting energy from food and building the molecules of life from scratch. It's the kind of detail that makes a biologist sit up a little. Not proof exactly, since a useful trick can always be borrowed later even if it wasn't there at the very beginning, but the kind of clue that's hard to entirely dismiss either. There's a second kind of vent, discovered rather later, in the year 2000, that some researchers find even more suggestive, though it's also more disputed, and it's worth being honest about that. These are called alkaline vents, and the most famous example, a field of white towers named Lost City, sits not on the fiery volcanic ridge where black smokers form, but off to one side of it, powered instead by a slower chemical reaction between seawater and a particular kind of deep mantle rock. The fluid that rises here is cooler than at a black smoker, gentle enough that you could, in principle, hold your hand near it without harm, and instead of gushing out as an obvious plume, it seeps quietly through a labyrinth of tiny connected pores and channels inside porous mineral chimneys, rather like water moving slowly through a sponge, or through the fine chambers of a sea cave carved by centuries of tide. Michael Russell, a geologist who had been thinking about this kind of chemistry since long before Lost City was ever found, argued that these pores might have served as something like ready-made cells, tiny compartments with mineral walls, each one bathed in a gentle, steady gradient between the alkaline vent fluid within and the more acidic ocean water outside. No membrane needed to be invented from scratch. On this view, the rock itself did that work for free, holding chemistry in place long enough for something more complicated to arise, compartment after compartment after compartment, a natural honeycomb built by mineral precipitation rather than by biology, waiting for the biology to catch up. It's a beautiful hypothesis, and no honest account should pretend it stands unchallenged. Some critics point out that vent water tends to be extremely dilute in the organic molecules life would need, and that any delicate, energy-poor building blocks forming in those tiny pores might simply diffuse away into the vastness of the ocean before they had any chance to concentrate or react further. Others question whether the particular chemistry proposed can really produce the more complex molecules, the sugars and nucleotides, that a working cell requires, rather than stalling out at some simpler intermediate stage. And a competing school of thought has argued for entirely different settings altogether: shallow pools on land, warmed and cooled by the sun in daily cycles, where wet and dry conditions might alternate in a way that concentrates chemistry rather than dilutes it. That disagreement hasn't resolved itself yet, and it may not resolve cleanly for some time. What the vents do offer, undeniably, is a working demonstration that energy, structure, and the right raw chemical ingredients can all show up together in the same unlit corner of the world, without any need for lightning or sunlight or a laboratory flask at all. And it's that particular kind of demonstration, chemistry finding a scaffold to work within, a boundary to lean against, that leads quite naturally toward a different question now, the question of what it would actually take to wrap all of this in a skin of its own. Let's set that question of skins and boundaries gently to one side for a moment, because before life could wrap itself in anything, it needed something worth wrapping. A boundary alone doesn't make a living thing. You could seal a raindrop's worth of ordinary seawater inside a soap bubble and wait 1,000 years, and nothing would happen inside it, because water and salt don't do anything on their own. Life needed a molecule, or a handful of molecules, capable of two very different jobs at once. It needed something that could hold information, the way a recipe holds information, so that a particular arrangement of parts could be preserved and passed along rather than lost to chance every time. And it needed something that could act, that could actually do chemistry, speeding up reactions and building structures, the way a pair of hands does the work that a recipe merely describes. For most of the history of modern biology, those two jobs seem to belong to two entirely different kinds of molecule. DNA, we were taught, holds the information. Proteins do the work. It's a tidy division of labor, and it maps neatly onto how a modern cell actually runs, with the double helix tucked safely away as an archive, consulted rather than touched, while proteins bustle around doing the actual chemistry, folding into their intricate shapes and catalyzing the reactions that keep a cell alive from one moment to the next. But that tidy division creates an uncomfortable puzzle when you try to imagine how life got started, because DNA cannot make proteins without help. It needs an elaborate reading apparatus, a small army of enzymes and structures, to translate its code into anything useful at all. And proteins, for their part, cannot be made without instructions, and more than that, the machinery that builds proteins is itself built largely out of protein. So you're left with something that looks uncomfortably like a locked door, with the key sitting on the other side of it. DNA needs protein to be read. Protein needs DNA, or something like it, to be specified. Which one, then, could possibly have come first, if each one seems to require the other already fully formed and functioning? The way out of that particular deadlock, first proposed seriously in the 1960s by a handful of scientists working somewhat independently of one another, among them Carl Woese, Leslie Orgel, and Francis Crick, of all people, the co-discoverer of the double helix itself, was to suggest that maybe neither molecule came first, because maybe there was a third molecule, quieter and less celebrated, that could do both jobs by itself. That molecule is RNA, ribonucleic acid, DNA's close chemical cousin, and in most textbook accounts of modern biology, it gets cast in a fairly minor supporting role, a kind of messenger boy, carrying copied instructions from the DNA archive out to the protein-building machinery, and then being quietly discarded once its errand is done. For a long time, that was assumed to be more or less the whole story of what RNA does. It copies, it carries, it gets recycled. Nothing more dramatic than that. Then, in the early 1980s, two researchers working separately, Thomas Cech at the University of Colorado and Sidney Altman at Yale, each stumbled onto something that upended that entire picture, and did so almost accidentally, in the way that some of the most important discoveries in biology tend to happen, not through a grand hypothesis being confirmed, but through a stubborn, puzzling result refusing to go away. Cech was studying a single-celled pond organism called Tetrahymena, investigating how a particular stretch of its RNA got trimmed and edited before use, a process that everyone assumed required a protein enzyme to carry it out, the way essentially every known biological reaction seemed to require a protein enzyme to carry it out. He kept trying to identify which protein was responsible, and he kept failing, because there wasn't one. The RNA was doing the trimming to itself. It was cutting itself with no protein assistance at all, snipping out a piece of its own sequence and stitching the remaining ends back together, entirely unaided. Altman, working on a different system involving an enzyme called ribonuclease P, found something closely related, an RNA component that turned out to be doing the essential catalytic work, while the protein attached to it was, comparatively speaking, just along for the ride, offering a bit of stability but not much else. This was a genuinely startling result at the time, startling enough that it took the wider scientific community a while to fully absorb what it meant, because it broke a rule that had seemed close to absolute. Catalysts, the molecules that speed up chemical reactions inside a cell without themselves being consumed, were assumed to be the exclusive domain of proteins. That assumption had a name almost as a matter of course, ribozyme not yet being a coined term, because nobody imagined they'd need one. Proteins had earned that role. Their long chains fold into elaborate three-dimensional shapes, full of pockets and grooves and precisely angled surfaces, each one specialized to grip a particular molecule and hold it in just the right position for a reaction to occur. RNA, by comparison, had always seemed like a much simpler, more limited kind of molecule, a fairly plain-looking chain that could pair with a complementary chain, and not much else. What Cech and Altman had found was that RNA could fold too, not quite as intricately as protein, but enough. Enough to create pockets, enough to position atoms precisely, enough to genuinely catalyze a reaction on its own. They called these catalytic RNA molecules ribozymes, a deliberate echo of the word enzyme, and in 1989, the two of them shared the Nobel Prize in Chemistry for the discovery. Once ribozymes were confirmed to exist, that old deadlock started to loosen its grip, because here, finally, was a single kind of molecule that could plausibly do both jobs at once. RNA can store information, using the same four-letter alphabet of chemical bases that DNA uses, arranged along a chain in a specific sequence that can be copied, in principle, from one strand to another. And RNA, as it turns out, can also fold into a shape and act as a catalyst, speeding along the very chemical reactions that a primitive cell would need to survive and to reproduce. A world built primarily around RNA, then, wouldn't require the deadlock of DNA needing protein and protein needing DNA. A strand of RNA could, at least conceivably, copy itself, or help copy other strands of RNA, using nothing but its own catalytic ability, and in doing so, it could begin to fulfill both roles that later life would split apart between two separate kinds of molecule. This idea came to be known as the RNA world hypothesis, a name suggested by the Nobel laureate Walter Gilbert in a short paper in 1986, and it proposes that at some early point, life, or something meaningfully approaching life, ran on RNA alone, before DNA and protein evolved later to take over the two jobs more efficiently. DNA prized for the chemical stability that lets information survive over long stretches of time, protein prized for the far greater versatility of its catalytic folding. It's a genuinely elegant idea, and it does real explanatory work, but it would be a mistake to hear it and imagine the puzzle as fully solved, because building an RNA world from scratch, without any life already present to help, runs headlong into some very stubborn chemistry. Individual nucleotides, the small building blocks that link together to form an RNA chain, are themselves difficult to assemble reliably under plausible early Earth conditions, and getting them to link up into a long, functional chain is harder still. RNA chains tend to fall apart about as readily as they form, especially in water, which happens to be the one environment early RNA would have needed to exist in. And even if a strand somehow assembled itself, copying it accurately, so that the information it carried could actually be passed on, rather than garbled into nonsense, is its own separate and substantial problem, one that modern cells solve using an elaborate suite of enzymes that plainly weren't available yet on a young, lifeless Earth. So the RNA world hypothesis doesn't hand us a finished answer so much as a much sharper question, a narrower target to aim at. If RNA really did come first, then somewhere in that ancient chemistry there has to have been a way for nucleotides to form, to link together into chains long enough to matter, and to be copied, however imperfectly, without any of the machinery that living cells now rely on to do exactly that. So set the RNA question aside for a moment, not because it's resolved, but because there's a second problem that has to be solved alongside it, and it's one that's easier to picture, even if it's no easier to explain. Imagine for a moment that somewhere in the shallow, mineral-streaked water of the early Earth, a short strand of RNA really did manage to form, and even to copy itself a few times over, however clumsily. What would happen to it next? Out in open water, with no walls around it, that strand would simply drift. It would spread out, dilute itself into the surrounding ocean or pond or pool, and any useful copies it made would drift away from it too, off to be useless somewhere else entirely, diluted past the point of meaning anything. A working set of molecules that can't stay together isn't much of anything, no matter how clever the chemistry inside it might be. For something to start behaving like life, it doesn't just need molecules that can store information and molecules that can catalyze reactions. It needs a boundary, some kind of skin, thin enough to let useful things in and keep waste products out, but sturdy enough to hold a little private chemical world together against the vast diluting patience of the ocean outside it. This is where membranes come in, and it turns out that membranes are, in some ways, the easiest part of this whole story to explain, because the molecules that make them practically build themselves. Picture a bottle of salad dressing left standing on a counter, oil pooled on top, vinegar underneath, a clean, stubborn line between the two. Oil and water simply do not want to mix, and no amount of stirring keeps them mixed for long, because water molecules are drawn to each other, tugging and tumbling around one another in a restless, clinging dance, and oil molecules have no way to join in. Left alone, the water molecules nudge the oil back out, back into its own separate pool, the two of them sorting themselves apart again as if by quiet agreement. Now imagine a molecule that is a little bit like both at once. A molecule with one end that loves water and clings to it eagerly, and another end that recoils from water the way oil does, wanting nothing to do with it. Chemists call the water-loving end hydrophilic and the water-fearing end hydrophobic, and molecules built this way, with a friendly head and an unfriendly tail, are called fatty acids, or more broadly, lipids. They are not exotic. They are, in fact, some of the easiest organic molecules to produce under early Earth conditions, and they've turned up again and again in the same meteorite fragments and laboratory simulations that produced amino acids, and in the warm alkaline vents at the bottom of ancient oceans, where the right minerals could nudge simple carbon compounds together into exactly this kind of two-faced structure. Drop enough of these fatty acid molecules into water, and something almost effortless happens. Each molecule wants its head in the water and its tail out of it, and with countless molecules jostling for the same arrangement all at once, they settle into it collectively, without any planning, the way a crowd of people all trying to keep their feet dry will naturally end up walking the same narrow strip of a dry sidewalk. If the molecules are floating loose in solution, they can cluster into a little ball called a micelle, with the water-loving heads facing outward into the water and the water-fearing tails huddled together in the dry, sheltered center, hidden away from the water on every side. But if the concentration is right, something more interesting happens instead. The molecules pair up, tail to tail, forming a double layer, a bilayer, with water-loving heads facing outward on both sides, toward the water inside and the water outside, and water-fearing tails sandwiched together in between, shielded from water entirely. Curve that double layer around on itself, close it into a sphere, and you get something remarkable. A hollow bubble with a wall exactly 2 molecules thick, sealed off from the water around it, and yet made of nothing more exotic than fatty acids arranging themselves according to their own simple preferences. Scientists call these bubbles vesicles when they're made in a laboratory under controlled conditions, and protocells when they're being discussed as plausible ancestors of the very first living cells, structures that could have assembled themselves, unplanned and unassisted, in the shallow pools and volcanic pores of the young Earth. It's worth sitting with how strange and how ordinary this is at the same time. A soap bubble is, in a rough sense, a cousin of the same idea, though soap is built a little differently, and its shell is really a thin film of water trapped between 2 layers of soap molecules rather than a sealed double layer enclosing water on the inside. Still, the basic instinct is the same one. Molecules with a friendly side and an unfriendly side, sorting themselves into the most comfortable shape available, and in doing so, without meaning to, drawing a boundary between an inside and an outside. That boundary is not a strong one. It is not built the way a brick wall is built, with the deliberate, engineered strength of something designed to keep an interior separate from an exterior come what may. It's closer to a loose crowd of people holding hands, cooperating to make a shape, but ready to let go and reform if the crowd is jostled hard enough. And that looseness turns out to matter enormously, because it gives these early membranes a property that modern cell membranes, with their far more elaborate and controlled machinery, had to specialize away from over enormous stretches of time. Modern cell membranes are picky. They're built from more complex lipids, and they're studded with specialized proteins that act like doormen, checking each molecule that wants to cross and letting through only the right ones, at the right times, often at real energetic cost to the cell. But a simple fatty acid membrane, the kind that could have assembled itself on the early Earth with no proteins and no doormen at all, is leaky in a useful way. Small molecules, things like amino acids, simple sugars, even short nucleotides, can slip through the gaps between the loosely packed fatty acid tails, diffusing in and out of the protocell almost as freely as they would move through open water, while somewhat larger molecules, like long RNA chains, are too big and too charged to pass through as easily, and tend to stay trapped inside once they've formed there. This is a genuinely wonderful piece of built-in chemistry, because it means an early protocell could have drawn in a steady trickle of small raw materials from its surroundings, the very ingredients needed to build RNA, sugars, energy-carrying molecules, while still holding on to the more complex, harder-won molecules it managed to assemble internally. The membrane didn't need a mechanism to know the difference. Size and shape did that work for free. And these protocells weren't necessarily static either. Laboratory experiments with fatty acid vesicles have shown that when more fatty acid molecules are added to the surrounding water, existing vesicles can actually grow, absorbing the new material into their own walls and swelling larger, sometimes stretching into long, thread-like, almost worm-shaped forms. And a vesicle stretched that way, thin and elongated, is mechanically unstable in a very particular sense. Gentle turbulence, the kind of everyday agitation you'd find from waves lapping against rock, or bubbles rising through a warm vent, or simply the wind stirring the surface of a shallow pool, can be enough to pinch a stretched vesicle apart into two smaller ones, each sealed, each independent, each carrying away a share of whatever had been drifting around inside the original. It is, in other words, something that looks uncannily like growth and division, the two most basic hallmarks of a living thing, and it doesn't require any biology to produce. It's just fatty acid chemistry and a bit of environmental jostling, doing on its own something that, once life existed to inherit and refine it, would eventually become one of the most tightly regulated and essential processes any cell performs. But here is where the story runs back into that same stubborn kind of problem it kept running into with RNA, because growth and division on their own don't guarantee anything meaningful is being passed along. If a vesicle divides and its contents simply get divided along with it at random, some RNA molecules ending up in one daughter bubble, some in the other, with no method to it at all, then division is really just dilution happening in slow motion, spreading whatever useful molecules exist ever thinner across an ever-growing population of separate compartments, rather than concentrating and preserving anything of value. So the lipid world story, for all its elegance, still needs something else layered on top of it before it becomes a full account of how life could have gotten started, some way of making division non-random, some way of coupling what's inside a vesicle to how and when it splits. And that admission that even the most physically satisfying part of the story has a hole in it is a good place to pull back and look at the whole landscape a little differently, because up to now it might have sounded as though there's one correct path being slowly uncovered, one sequence of events that happened on the early Earth, and that scientists are gradually reconstructing detail by detail, the way you'd reassemble a shattered pot from its shards. That isn't quite the right picture. What actually exists is more like a set of overlapping research programs, each one favoring a different starting point, each one good at explaining certain things and weak at explaining others, and each one pursued by researchers who are often perfectly aware of the other's strengths and quietly trying to borrow from them. The lipid world idea, which says that self-assembling fatty membranes came first and gave chemistry a place to concentrate and experiment, is one such program. the RNA first idea from earlier, which says that a molecule capable of both storing information and doing chemical work must have come before anything else, because otherwise you need chicken and egg simultaneously, is another. But there are several more, and it's worth walking through them slowly, not to decide a winner, but to get a feel for how differently the same deep question can be approached. Consider what's sometimes called the metabolism first view. Instead of starting with a special molecule, whether RNA or a fat, this approach starts with chemical reactions themselves, with cycles of transformation, one substance becoming another, and eventually looping back around to something resembling the first, releasing or capturing energy at each step, the way your own cells still run cycles today. Small molecules shuttled through repeating loops of chemical change that keep the whole system running. The appeal here is that you don't need to explain the sudden appearance of a complicated polymer like RNA. You only need simple molecules and a source of energy, something like the mineral surfaces and chemical gradients found around deep-sea hydrothermal vents, where hot, mineral-rich water, rich in hydrogen and other reactive substances, pushes up through tiny pores in rock and meets colder ocean water, creating exactly the kind of steady chemical imbalance that a primitive metabolism might have needed to get going, the way a water wheel needs a stream flowing downhill rather than a still pond. Some researchers who favor this picture point out that the pores themselves, in certain kinds of vent rock, are already roughly cell-sized, tiny mineral chambers a fraction of a millimeter across, naturally structured almost like a honeycomb, which would have offered something membrane-like without needing any membrane at all, at least at first. The weakness, of course, is the same one that dogs every metabolism first idea. A cycle of chemical reactions, however elegant, doesn't obviously store information or pass anything down to a next generation, and without that, it's hard to see how such a system could improve itself over time rather than just running in place indefinitely. Then there's what's sometimes called the peptide first view, which starts not with RNA or fats or metabolic cycles, but with proteins, or rather with the short chains of amino acids called peptides that are simpler than full proteins, but still capable of folding into shapes and catalyzing certain reactions. Amino acids, after all, are some of the easiest biologically relevant molecules to make from scratch. They turn up reliably in spark discharge experiments. They've been found in meteorites that fell to Earth decades ago and were quickly retrieved before contamination could set in, and they link together into peptide chains under surprisingly mild, plausible early Earth conditions, particularly on certain mineral surfaces that seem to encourage amino acids to bond and lose water in the process. The trouble is the same one you'd expect. Peptides are wonderful at catalysis, at speeding up and shaping reactions, but they don't have an obvious way of storing and copying information the way RNA's sequence of bases does. So a peptide-first world needs some other explanation for how heredity eventually entered the picture. There's also a whole line of thinking built around clay, ordinary clay minerals, the kind you'd find as slick mud along a riverbank. Certain clays, it turns out, have surfaces made of layered, repeating crystal structures with regularly spaced charged sites, and laboratory work has shown that these surfaces can attract small organic molecules, hold them in place, and even help line them up in ways that encourage them to link together into longer chains, functioning almost like a scaffold or a template. Some researchers have gone further and suggested that the very first replicating structures weren't organic molecules at all, but the clay crystals themselves, imperfections in their layered structure, copying themselves as the crystal grew. A strange and admittedly speculative idea, but one that at least offers a concrete answer to where a template for copying might have come from before biology existed to build one. It's a minority view within the field, and most researchers treat it as more of a thought experiment than a leading hypothesis, but it's a useful reminder of just how many possible on-ramps there are into this problem. Beyond the ingredients themselves, there's disagreement about the setting, too. The hydrothermal vent picture, favored by many metabolism-first researchers, imagines life beginning in the ocean depths, in darkness, warmth, and a steady chemical gradient. But a different camp of researchers has spent decades building the case for shallow terrestrial hot springs instead, the kind you might find in a volcanically active landscape, bubbling pools ringed with mineral deposits, similar in spirit to the geothermal features found in places like Yellowstone today. The argument for this setting has less to do with the pool's warmth and more to do with what happens at their edges, because a shallow pool that periodically dries out, exposing its rim to sun and air before refilling with the next rain or the next surge from underground, puts any molecules trapped there through repeated cycles of wetting and drying, concentrating them, forcing them into contact, and in some laboratory versions of this process, encouraging exactly the kind of bonding that links small building blocks into longer chains, an effect that's much harder to arrange at the bottom of the ocean, where everything simply stays dissolved and dilute. And then, almost in the opposite direction entirely, there's a body of work exploring what would happen if the early Earth, or at least parts of it, were considerably colder than commonly imagined, cold enough that stretches of ocean or fresh water lay frozen for long periods, with liquid water surviving only in thin channels and pockets threading through the ice, channels that happen to concentrate dissolved molecules remarkably well as the surrounding water freezes around them, the same way a puddle left to freeze slowly will often push its dissolved minerals into the last remaining liquid at its center. Cold conditions also slow down the breakdown of fragile molecules like RNA, giving them more time to accumulate and react before falling apart, an advantage that a warm, sunlit hot spring simply doesn't offer. None of these programs, the metabolic, the peptide, the mineral, the vent, the spring, or the ice, has managed to explain the whole transition from chemistry to biology entirely on its own, and that's precisely why so much current work has started leaning toward hybrid scenarios instead, ideas that borrow a piece from each, perhaps membranes from one setting, information-carrying molecules synthesized somewhere else and later transported in, catalysis, contributed by peptides, energy supplied by mineral surfaces, with wet-dry cycling somewhere in the mix to drive polymerization forward. It's less a single sweeping origin story than a patchwork being stitched together from many directions at once, each thread tested separately in laboratories around the world, none of them yet fully joined. And it's worth sitting with that patchwork for a moment, because the next question worth asking isn't which single setting is correct, but what it would even take for any of these separate ingredients, once they existed side by side, to start behaving as though they belonged together. Belonging together, in the strict biological sense, means something quite particular. It doesn't just mean sitting near one another in the same warm pool or the same mineral pore. It means that whatever happens to one molecule changes the odds for the others around it, that helping a neighbor copy itself, however crudely, comes back around to help you too. That's the seed of selection, and it can start operating long before anything resembling a cell exists, long before there's a membrane to call a boundary, or a nucleus to call a control center. All selection really requires, at its most bare bones, is variation, replication, and some difference in how well the variants persist. If a population of molecules includes some that copy themselves a little more accurately, or a little more quickly, or a little more resistant to falling apart in the heat of the day, those variants will tend to become more common over time, purely as a numbers game, without anyone or anything intending it. The chemist Manfred Eigen, working in Germany in the 1970s, spent a great deal of time thinking through what this kind of molecular selection would actually look like in practice, and he ran into a problem that turns out to be central to almost every serious theory of life's origin. He called it the error threshold, and the idea, once you sit with it, feels almost obvious in hindsight, the way the best insights often do. Imagine a molecule capable of copying itself, say an early RNA strand, but copying itself imperfectly, the way any chemical process without a proofreading system will inevitably be imperfect. Every time it makes a copy, there's some chance of a mistake, a letter swapped, a piece dropped, a stray base inserted where it doesn't belong. If the molecule is short, a handful of building blocks strung together, a single mistake here or there might not matter much. The copy is close enough to the original to still function, still fold into roughly the same shape, still do roughly the same job. But as the molecule grows longer, as it accumulates more information, more instructions, more complexity worth preserving, the odds that any single copying event will produce an error somewhere along its length go up correspondingly, and at some point, a threshold is crossed. Beyond a certain length, for a given error rate, the copies drift away from the original faster than selection can hold them in place. The information essentially dissolves back into randomness, generation after generation, like a whispered sentence passed down a long line of people, each one hearing it slightly wrong, until what comes out the other end bears no resemblance to what went in. This creates what's sometimes called a catch-22 for the origin of life, though it isn't quite as hopeless as that phrase makes it sound. You need a fairly long, fairly complex molecule to build an accurate copying machine, something sophisticated enough to check its work and correct mistakes as it goes. But you can't reliably build and preserve that long, complex molecule without already having accurate copying in place. It's a bit like needing a printing press to manufacture the parts for a printing press. Modern cells solve this problem with an elaborate toolkit of enzymes dedicated entirely to proofreading, catching errors, and repairing damage. A whole quality control department humming away inside every dividing cell. But that toolkit itself had to come from somewhere, and it couldn't have existed at the very beginning, when nothing resembling an enzyme yet existed to do the proofreading. One way around the problem, and this is where cooperation starts to matter, is for the job of holding information to be spread across several shorter molecules instead of piled into one long one. A short RNA molecule can be copied with reasonable fidelity even by a sloppy, primitive process, because there's simply less of it to get wrong. So instead of one strand carrying everything, imagine a small community of short strands, each one responsible for a different task, each one below its own individual error threshold, but together, capable of doing something none of them could manage alone. Eigen and his colleague Peter Schuster proposed a version of this idea they called the hypercycle, a set of molecules arranged so that each one helps catalyze the copying of the next, in a loop, so that the whole cycle reinforces itself. It's a cooperative structure, and cooperative structures bring their own headaches, because cooperation is always vulnerable to cheating. A molecule that stops contributing to its neighbors, that simply gets copied without paying anything back, will often out-reproduce the more generous molecules around it, the same way a hitchhiker who never buys gas can travel more cheaply than everyone chipping in for fuel. Left unchecked, the cheaters tend to spread, and the whole cooperative system can collapse under its own selfishness, a genuinely serious hazard for any theory that leans on hypercycles. This is very likely part of why membranes matter so much, and why so many origin of life researchers keep circling back to compartments even when their favorite chemistry happens somewhere else entirely. A membrane, even a leaky primitive one made of simple fatty molecules, does something quietly powerful. It keeps a molecule's cooperative partners nearby, prevents the useful products of one strand's activity from drifting off to benefit some other strand's freeloaders in the next pool over, and it lets natural selection act on whole packages rather than isolated molecules. A vesicle whose contents happen to cooperate well will grow and divide more successfully than a vesicle whose contents are riddled with cheaters, and so selection at the level of the compartment can start to police the selfishness happening inside it, favoring the vesicles that hold their cooperative arrangements together. It's an early, chemical version of a lesson that shows up again and again throughout the history of life, that cooperation, once it finds a way to protect itself from exploitation, can outcompete pure self-interest. Somewhere in the midst of all this experimenting, sorting, and slow accumulation of workable arrangements, three particular kinds of molecules seem to have found their way into a shared arrangement so useful, so mutually reinforcing, that it eventually became the foundation for essentially all life on Earth. Those three are the nucleic acids, RNA and eventually DNA, which store information, the proteins, built from amino acids, which do most of the actual physical work inside a cell, folding into shapes that grip, bend, and catalyze, and a peculiar molecular machine called the ribosome, itself built largely out of RNA, whose job is to read the information stored in nucleic acids and use it to build proteins according to that stored recipe. The system that connects these three, translating a sequence of genetic letters into a sequence of amino acids, is what biologists call the genetic code, and one of the more striking features of that code is that it appears to be, with only minor variations, the same code used by essentially every living thing on the planet, from the bacteria in a drop of pond water to the cells lining a human's own throat. That universality is itself a clue, suggesting the code was locked in place very early, before life diversified into its separate branches, and has simply been inherited ever since. The way a founding decision made generations ago can end up shaping every institution that grows from it long afterward, whether or not the decision still makes the most sense in hindsight. What makes this arrangement genuinely puzzling, rather than simply intricate, is that each piece seems to depend on the others in ways that are hard to unwind. Proteins are needed to build and copy nucleic acids efficiently in modern cells, but nucleic acids are needed to specify which proteins get built in the first place. The ribosome that translates the code is itself made largely of RNA, and yet it's also, in a very real sense, a kind of ancient fossil, a molecular machine that may be quietly preserving evidence of a moment before proteins had fully taken over the roles RNA once played on its own. That fossil metaphor is not just poetic license. Researchers who study the ribosome closely, mapping its oldest and most essential regions, have found that the parts responsible for actually forming the chemical bond between two amino acids are made entirely of RNA, with no protein involved at all in that specific step. The proteins scattered around the ribosome seem to be later additions, scaffolding bolted onto a core structure that was doing its essential work long before proteins arrived to help stabilize and refine it. It is a little like discovering that a modern office building, glass and steel and elevators, still has at its center a much older stone foundation, load-bearing and irreplaceable, that nobody thought to remove when they built everything else around it. The tenants come and go, the furniture changes with every decade, but the foundation keeps doing the one job it has always done. This is the kind of clue that origin of life researchers live for, because it lets them work backward from evidence rather than simply speculating forward from possibility. And in the last few decades, this work has moved substantially out of the realm of pure argument and into laboratories, where people actually try to build the intermediate steps with their own hands, using glassware, temperature-controlled water baths, and a great deal of patience. One of the more influential of these efforts has centered on a chemist named John Sutherland, working in Cambridge, England, who spent years trying to solve a problem that had frustrated origin-of-life chemistry for decades. Nobody could get the building blocks of RNA to assemble themselves efficiently from simple starting materials, no matter how they mixed them. Sutherland's insight, which took most of a decade of failed attempts to arrive at, was that instead of trying to build the sugar and the base separately and then join them together, as life does today, the earliest chemistry might have built the whole nucleotide as a single connected structure from simpler pieces, using ultraviolet light from the sun to help purify the mixture by destroying unwanted side products. It was, by his own account, a discovery that came only after abandoning an assumption everyone had been quietly making, the assumption that a plausible early Earth pathway needed to closely resemble the way modern biology assembles its own molecules today. Once he stopped requiring that resemblance, new possibilities opened up that had been invisible before. Elsewhere, in a lab at Harvard, a biologist named Jack Szostak has spent much of his career on a different piece of the puzzle. Not how the letters of RNA might have formed, but how the earliest compartments, the ancestors of what would become cells, might have assembled themselves without any biological machinery to guide them. His approach uses fatty acids, simple oily molecules that, when placed in water, spontaneously arrange themselves into tiny hollow spheres, essentially soap bubbles built from a single layer or two of molecules rather than water and air. Left alone, these little spheres will grow by absorbing more fatty acid material from their surroundings, and under the right gentle physical stress, such as being pushed through a narrow space or agitated slightly, they will divide into daughter spheres, a crude, chemical version of the reproduction that every living cell performs today, no genes or proteins required. Szostak and his collaborators have shown that these protocells, as they're often called, can encapsulate RNA molecules inside themselves and, under laboratory conditions, allow those enclosed molecules to be copied, using nothing but the chemistry of nucleotides, sticking to a template strand, and occasionally, rarely, linking up on their own. It is slow going, and nothing built this way in a laboratory has yet crossed the threshold into anything a biologist would confidently call alive. But each piece, the compartment that holds itself together, the RNA that carries information, the copying process that could in principle allow that information to be inherited, gets a little more convincing every few years, and the gaps between them get a little narrower. None of this laboratory work claims to have recreated the actual history of the origin of life on the early Earth, and the scientists doing it are usually the first to say so. What they are testing instead is plausibility. Could chemistry alone, without any guiding hand, cross from simple molecules to something that behaves, even faintly, like life? Each experiment that succeeds narrows the space of things that still need explaining. Each experiment that fails, and there have been many, tells researchers which assumptions to abandon and try again from a different angle, the way Sutherland's team eventually did. While these chemists work at their benches, another kind of search has been unfolding much farther away, on the actual surfaces and beneath the actual ice of other worlds. Mars has drawn particular attention, not because anyone expects to find something obviously alive crawling across its red dust, but because billions of years ago, before Earth's own origin story had likely finished playing out. Mars appears to have had liquid water on its surface, a thicker atmosphere, and conditions that might, for some stretch of time, have resembled the young Earth closely enough to run the same experiment in parallel. Robotic rovers, including one currently working its way across an ancient river delta inside a crater called Jezero, have been drilling into rock and sealing away samples specifically chosen because they formed in what was once standing water, samples that a future mission is intended to eventually bring back to laboratories on Earth, where instruments far more sensitive than anything that can be flown to Mars could examine them for the chemical traces that ancient biology, if it ever existed there, might have left behind. Farther out still, past Mars and the asteroid belt, several of the icy moons circling Jupiter and Saturn have become unexpectedly serious candidates in this search, for a reason that would have seemed strange to earlier generations of scientists who assumed that a place needed a warm sun and open sky to support anything living. Moons like Europa and Enceladus are covered in miles of solid ice, but underneath that ice, warmed not by sunlight but by the flexing and squeezing of powerful tidal forces from their parent planets, both appear to hide entire oceans of liquid water, sealed away from the vacuum above. At Enceladus, spacecraft have actually flown directly through plumes of water vapor and ice crystals erupting from cracks near its south pole, sampling material from that hidden ocean without needing to drill through a single inch of ice, and finding within it the kinds of simple carbon-based molecules and mineral chemistry that, on Earth, tend to accompany the sort of hydrothermal vent environments some scientists suspect were involved in life's own beginning. If life, or even just the chemical stepping stones toward life, were ever found on Mars or in the ocean of Enceladus, it would be one of the most significant discoveries in the history of science. But it is worth being honest, even in a moment as quiet as this one, about exactly what such a discovery would and would not settle. Finding evidence of life that arose completely independently on a second world would answer a question that has hung over this entire subject for centuries: whether the origin of life is a routine outcome of ordinary chemistry, something that happens reliably wherever the right ingredients and conditions come together, or whether it is instead a wildly improbable accident, one that might have happened only once in the entire observable universe, here, on this one damp and unremarkable planet. A second independent origin, even a simple one, would suggest the universe is far more generous with life than a single data point could ever prove on its own. But it would not, by itself, tell us which particular pathway Earth's own life actually followed, whether RNA truly came first here, or clay surfaces, or some other chemistry lost entirely to time. Discovering life elsewhere would open a second window onto the same fundamental mystery, not close the one we've been looking through all along. Even with everything we've walked through together, from the forging of carbon inside dying stars, to Miller and Urey's sparking flasks, from clay surfaces that might have lined up the first fragile chains of RNA, to the alkaline chimneys of the deep-sea floor, there remains a genuine and honestly quite large gap in the story, one that no laboratory has yet closed. It sits right in the middle, between simple chemistry doing something clever and a true cell doing something alive. We can make nucleotides link into short chains under the right laboratory conditions. We can make fatty molecules spontaneously curl themselves into tiny hollow spheres, something like soap bubbles, that look uncannily like primitive membranes. We can even coax some short RNA strands to copy short RNA strands, slowly and imperfectly, without any help from proteins at all. But nobody has yet taken all of these separate, promising pieces and assembled them into something that can sit in a beaker, take in raw materials from its surroundings, use energy to build more of itself, and pass a set of working instructions onto something that qualifies as an offspring. That handoff, from chemistry that merely behaves in interesting ways to chemistry that reproduces itself with enough fidelity for evolution to start acting on it, is the part of the story that remains, even now, essentially unfinished. Part of the difficulty is that so many of the pieces seem to depend on each other in a way that makes it hard to know which one showed up first. A cell needs a membrane to hold its contents together, so that useful molecules don't simply drift away into the surrounding water the moment they're made. But a membrane on its own is just a bag. It needs some kind of internal chemistry, some way of using energy, in order for anything interesting to happen inside it. And that internal chemistry, in every cell we know of today, is directed by genetic instructions, sequences of RNA or DNA that specify which molecules to build and when. Yet, those genetic instructions are, in modern cells, built and read and copied by an elaborate cast of protein machines, and those proteins are themselves built according to instructions carried in the genes. It's a bit like trying to figure out which came first among a lock, a key, and the locksmith who knows how to cut new keys from old ones. When in the story, you're trying to reconstruct, all three seem to need each other to exist at all. Researchers have proposed various ways this circle might have first been broken, various simpler arrangements that could have bootstrapped the whole system before it became this tangled. But none of these proposals has yet been demonstrated well enough, or agreed upon widely enough, to be called an answer rather than a promising direction. There's also the plain fact that we are trying to reconstruct an event that left almost no direct physical trace. The oldest rocks that might carry hints of early life are so old and have been so thoroughly cooked and folded and reshaped by billions of years of geological churn that even the most careful geochemists disagree about whether particular carbon signatures in them are truly biological or might have formed through some overlooked non-living process. By the time we reach the earliest organism we can describe with real confidence, the one biologists call the last universal common ancestor, life was already fully modern in its essential machinery. It already had DNA, a genetic code, ribosomes for building proteins, and a functioning metabolism. Everything that happened before that point, the whole span of chemistry we've spent this long journey exploring together, has to be inferred rather than observed directly, pieced together from chemistry experiments, comparisons between living organisms, and geological clues, the way a detective might reconstruct an evening from a handful of scattered, incomplete receipts, rather than from a security camera that was actually running the whole time. This is worth sitting with honestly, rather than smoothing it over, because it's easy for any account of life's origin, including this one, to make the story sound tidier and more settled than the actual science currently allows. What can honestly be said is that scientists have identified plausible chemistry for many of the necessary ingredients and plausible environments where that chemistry could have occurred, and plausible general pathways by which those ingredients might have come together into something self-sustaining. What cannot yet be said is that any single laboratory result stands as proof of exactly how it happened on the early Earth, on some particular afternoon, in some particular pool of water or crack in the sea floor 4 billion years ago. The honest position held by most serious researchers in the field is that the origin of life is an open scientific question with several live and competing hypotheses being tested and refined by real ongoing experiments rather than a solved mystery being merely explained after the fact. And yet, whatever the unresolved details, something in this story is not in question at all, and it is, in its own quiet way, worth carrying with you as you drift off tonight. The atoms sitting in your body right now, the carbon in your fingernails, the nitrogen in the proteins folded inside your muscles, the phosphorus holding together the very DNA curled up inside nearly every one of your cells, did not appear when you were born. They are, every one of them, older than the sun. They were cooked inside stars that lived and died long before our solar system ever gathered itself out of a spinning cloud of dust and gas, and they have been recycled ever since, through comets and asteroids, through the cooling crust of a young and violent Earth, through ancient oceans and tidal pools and mineral-laced cracks in rock. At some point, in some environment we still can't fully specify, some collection of these very old atoms crossed a threshold, and simple chemistry became something that could hold on to information, use energy, and make more of itself. From that first crossing, an unbroken chain of copying has run continuously forward, cell dividing into cell, for something like 4 billion years, never once stopping completely, all the way to this very evening, to the particular arrangement of that ancient chemistry that happens, for now, to be you, lying still, breathing slowly, listening. There is something quietly steadying in that fact, whatever remains uncertain about the exact mechanism. You are not separate from the process this whole story has been describing. You are, in a very literal chemical sense, still that process, still that same conversation between carbon and water and energy that began somewhere in the deep, dark, patient chemistry of the early Earth, still running, still being copied forward, cell by cell, breath by breath, tonight and every night. The scientists still working on the unanswered parts of this puzzle, the ones testing new versions of the RNA world, the ones studying hydrothermal vents and icy moons, the ones arguing over which came first, the membrane or the metabolism or the genes, are not chasing some abstract question far removed from you. They are trying to understand the deep history of exactly the chemistry that is, at this very moment, quietly keeping your heart beating and your lungs filling with air, without you needing to think about any of it at all. So let the questions stay open, the way they honestly still are, and let your body keep doing what it has been doing, uninterrupted, since long before there were minds able to wonder about it. The chemistry doesn't need you awake to keep working. It never has. Rest well.